I think so. It's 8:01. Okay. Good evening, everyone. Thank you for joining us. I'm Felicia Morton, the Executive Director and founder of the CGD Association of America. We are really pleased tonight to bring to you a very special event. This is something that everyone's been talking about. It's a topic on everyone's minds, and it has been for many years now, gene therapy for CGD. I know that when I was in the position that many of you are, thinking about what you're going to do for yourself or for your child, it was on my mind when I was a mother and considering options. And it was always something that was around the corner. Well, we're around the corner. The time has come. There are opportunities now. There's a clinical trial that has started. And there are a lot of questions that we have. And there are a lot of people tonight who have questions. I think we have maybe a record-breaking audience tonight. 50 people. I think more than 50 people actually now have joined this call, which is a testament to how important this topic is and the work that we're doing here at the CGD Association of America. We are growing, and it's because of you. It's because of our community and how strong we are. Our voices are being heard. And I want to do a little housekeeping at first because we have a big crowd tonight. So I want to make sure that everyone knows that you are on mute, but I've asked you to be on mute when this started, but we'll have a Q&A at about 8:35 where you can ask Dr. Leiding your questions. Until then, we will have all of our images on mute, and then we will also be able to ask you to unmute to ask your questions. Also, this call is being recorded. As you know, this was something that we published, and you saw it when you signed on. If you have any desire to turn off your video or not have your name, please do so. And in terms of some other items, we had talked about how we're growing. We really look forward to joining us on all of our social media platforms. For those of you who have not joined us on Instagram, please do. That's where you find out about everything that we're doing, these Zoom calls, different events that we're having, all the news and updates, anything that's going on in the CGD world and beyond. That's where you're going to get it at Instagram, on Facebook. We even started Snapchat. We're on TikTok. We're everywhere you want to be. So please follow us now if you haven't already. And Colton, our trusty consultant here, he is going to put our Instagram and some of our handles in the chat so that you know where to find us. And we also ask you to always sign up if you haven't already and be involved in our online chats and newsletter as well. So without further ado, I want to share why we're here today, and that is to talk about gene therapy with one of our top CGD experts in the world. And we're so grateful that she is also on our CGDAA Medical Advisory Board. And she has joined us here tonight to talk about this topic that's on everyone's minds. It's Dr. Jennifer Leiding. She is with Johns Hopkins All Children's Hospital in St. Petersburg, Florida. She is the Associate Director for Clinical Research in the Institute for Clinical and Translational Research and Assistant Director of the Center for Cellular and Gene Therapy in the Cancer and Blood Disorders Institute. She is also an Associate Professor of Pediatrics in the Johns Hopkins University School of Medicine. We're so grateful that you could join us here today, or tonight rather, Dr. Leiding, and we have your presentation loaded, and we look forward to hearing what you have to tell us about the latest in gene therapy for CGD.
Well, thank you, Felicia, and thank you for the warm welcome and for everybody who's in attendance. Maybe we should just get started and have lots of time for discussion, hopefully. So Colton is bringing up slides. Are you able to put it kind of in a presentation mode, Colton? Or like, you know what I mean, the slide share? Yeah, there you go. Perfect. All right, we can skip the title slide with all my name and everything. Just a couple of disclosures. So I do have some consultancy roles and research funding. I also worked for a gene therapy company previously, but that ended over a year ago and has nothing to do with what I'm going to talk about now. All right, next slide. Correct. I always like to start talks with CGD just to remind people what CGD is. Obviously, this audience knows exactly what CGD is, but it really matters from a genotype perspective. And so I just want to level set so we all are using the same vocabulary as we kind of move through this presentation. So just to review, so CGD is due to defects in any of the components of the NADPH complex, and that picture is being shown here where the X is over GP91 FOX and P22 FOX. There are now 6 different genes that cause CGD. It used to be 5, but now we have a new one on the block. And patients develop recurrent or severe infections with a subset of specific bacteria and fungi. Patients also have a really high rate of inflammatory disease and autoimmune disease, especially of the GI tract, lungs, and liver. And these become really important characteristics when we start thinking about clinical trials and how to measure disease activity. So the 2 that are going to become the most relevant in this conversation, and just to kind of point out, are GP91 FOX, which is that yellow square, which is the cause of X-linked CGD, and then P47 FOX, which is the green circle and is the most common cause of recessive CGD. All right, next slide. So in addition to gene therapy, I am going to talk a little bit about transplants. I can't not talk about transplants when we talk about curative options for CGD, because stem cell transplant is still a viable option and something patients should certainly consider when they're thinking about curative options. But when do we as doctors actually think about it? It might be different from when you guys as patients and advocates think about it. But as from a medical perspective, we start thinking about transplant for severe life-threatening infections, which can happen as early as birth or in infancy, the development of inflammatory disease, specifically colitis, granulomatous lung disease, but really any of them on the list could fulfill this criteria. If a patient has a mutation in one of these genes, oh, you got to go back, Colton.
Sorry, I was trying to admit someone.
It's okay. If a patient has, ooh,
One more?
No, forward one, please.
Oh, forward one.
Sorry, my child just got home. A mutation that predicts really high mortality. So physicians or the medical team may think of elective transplants or elective curative options in this instance, where we know that the mutation portends a worse prognosis and want to intervene earlier before the development of disease-related sequelae. And then it really can be, we're now getting into the field of elective transplantation or even gene therapy, where patients just want to seek curative options even before the development of these problems. All right, next slide. So one of the first studies that we did, I'm part of the Primary Immune Deficiency Treatment Consortium, which is a large consortium made up of 47 institutions in the United States, and really studies the natural history of chronic granulomatous disease, or CGD, and interventions and treatments for it. And so one of the very first studies that we ran in this consortium was trying to look at whether transplant actually resolves some of the problems of CGD that historically were not necessarily felt to potentially be cured by transplant. And in this case, we looked at inflammatory bowel disease, and we were able to show that presence of inflammatory bowel disease does resolve with transplant, and it doesn't worsen survival. So having IBD does not change the outcome of the transplant, but is a reason to be transplanted because it can be cured by the transplant itself. And this is IBD, obviously related to CGD. All right, next slide. And so this led to even more data that's come out. So Europe and the US are always competing with each other in regards to our CGD transplant outcomes, and they often beat us, unfortunately. But looking at this study, over 700 patients in Europe were enrolled on a study just looking at outcomes of transplants. And so in this case, they looked at overall survival, which was around 85% following transplant for CGD, and event-free survival, which was around 76%. Now, what event-free survival means is that you've survived the transplant without an event. And the study team defines that event. And in most cases, those events include death, failure, so failure of the transplant working, or the development of graft versus host disease, which is a really unwanted side effect of transplant. And so in this case, the event-free survival, so getting to that time point without one of those events, was around 75%. And around 12% of patients in their cohort experienced graft failure. So what that means is the patient received the transplant, They went through the whole regimen, the whole caboodle, and it didn't work. The transplant failed. It either didn't take or it did take and it was quickly lost. And so you're right back to where you started. Return of disease, but you've been exposed or the patient's been exposed to the toxicity of chemotherapy. So that's obviously not at all what anybody wants. I also always point out that the survival is amazing. 85% is wonderful. It's great odds. It's really, really good following transplant, but it's not 100. So in our hands, when we get to around 80%, that's 1 in 5 people who are going to die from transplant. So that's not negligible. And that's what we talk about when we're giving people sort of the odds of coming through this. All right, keep going. Okay, so this is now looking at our data from PIDTC with a little bit smaller number of patients, but 240 patients that were enrolled between 1996 and 2018. And survival in our hands, so this is in North America, was around 82% with the event-free survival of about 69%. These were not statistically significantly different from the European data, so I'm not advocating that everybody go to Europe for a transplant, but they're pretty much in line with each other. Keep going. And what we were able to show, can you click one more time, Colton, was that the performance status of a patient, so that really is a quantifiable measurement of how sick a patient is. The better a patient is going into this transplant, i.e. meaning not ill, not sick, the better survival they will have. And that's what the score is. But we were able to show that the type of chemotherapy that a patient received, the type of matching that they received, the type of stem cell source, whether it was bone marrow or cord blood, did not impact survival. It was really how well they were going into the transplant was what dictated it. Next slide. We were also able to show that infection and inflammatory disease were not associated with survival. So patients who had history of infection or even active infection at the time of transplant or inflammatory disease, including uncontrolled inflammatory disease, did not impact that survival post-transplant. So patients going into those transplants with those disease-related symptoms didn't have worse survival. Next slide. Yep, you can skip that. And this was just showing that there were no other relations. I think I already have shown some of these actually, looking at the performance stats. You can skip this, Colton. Sorry, it's redundant. But like the European group, we also saw a high rate of graft failure or observed a high rate of graft failure, around 18%. So again, we're getting close to that 20 number of 1 in 5, roughly, patients are going to have graft failure following their transplants. And in our hands, we were able to attribute it to certain types of chemotherapy regimens, which we now recommend avoiding so that there's not this increased risk of graft failure. Next slide. So PIDTC was also able to show that our patients with recessive disease, those with P47 FOX deficient CGD, who are largely thought previously to not be candidates for transplant, will benefit from transplant. Go to the next slide. And so this paper showed that the 5-year overall survival was similar to our overall cohort of 85%, event-free survival of 77%. And the conclusion of this paper being that transplant can effectively alleviate the disease burden caused by P47 CGD, and therefore transplant or some other curative option should be considered in this patient population that historically was not necessarily pursuing these options. Next slide. Folks in PIDTC are also looking at alternative options. So we start getting into the world of patients who don't have great donor options. And so that includes things like cord blood transplant, mismatched unrelated donor transplant, and haploidentical transplant. And so this paper was put together by Danny Arnold at the NIH and Suhag Parikh, who's at Children's Hospital of Atlanta. And we're able to show that cord blood transplant is a viable option, although be it survival is about 10% lower than with other types of transplant for CGD, around 73%. And there was a higher rate of graft failure in this group than in the overall group. And so isn't necessarily the first choice for our type of transplant, but certainly can be utilized. And this paper is been accepted for publication, but is not yet out. All right, next slide. And lastly is looking at haploidentical mismatched unrelated donor transplant for CGD. So on the left side of the slide is the European data. Their paper was just published, showing really actually excellent survival with a haploidentical donor. And what a haploidentical donor is really a half match. So this is someone like a mom or a dad, or even a sister or sibling who's not a full match, who is being used as the donor for the transplant. And in their hands, they recommend the use of haploidentical donors when there's not other options, but do recommend these special graft manipulation techniques. And then lastly is our data. This is the PIDTC data on the right, showing sort of similar data that use of mismatched unrelated donors or mismatched related donors are viable options as well. And this data has been submitted to the tandem meetings for transplant that occur in February. So we'll see if it gets accepted, but it's hot off the press. All right, next slide. All right, I'm going to dive into gene therapy now. So challenges and how are we solving them? So the biggest challenges for transplant for CGD, other than finding a matched donor, which is really problem number one, is the development of graft versus host disease, how to prophylax it, how to prevent it, and ways that we solve that is with graft manipulation. The other major problem is graft failure, and we think we can attribute this to pre-transplant inflammation that many trials are now trying to blunt adequately to hopefully lead to improved kinetics of immune reconstitution. Next slide. And this is just a very busy slide showing some of the graft manipulation techniques that different physicians, different groups will use in order to achieve or reduce the frequency of graft versus host disease and achieve higher frequency of graft engraftment and less graft failure. Next slide. All right, so let me dive into gene therapy. So I saw the slide on social media like forever ago, and I stole it because I love it. And so this is not updated at all, but it's really showing that gene therapies are crossing every discipline, every pediatric discipline, every adult discipline, and they're here to stay. And so this is a technology that really can be leveraged to treat patients with many, many, many different diseases, especially CGD, which is why we're here today. But this is really just showing the number of companies that are pursuing approvals from the FDA for gene therapy in various stages of clinical development using various types of technologies. Next slide. So limitations about why would we think about gene therapy in the first place, other than it sounds cool. So there's definitely limitations of allogeneic transplant, also limitations of gene therapy. But if we look on the left side, some of the allogeneic transplants limitations include a lack of well-matched donors. So if you are a patient who does not have a well-matched donor, you're left with using cord blood, if you can even find that, or a mismatched or a haploidentical donor, which have their own challenges associated with them. There's a risk of primary graft failure or death associated with it. It requires 2 to 3 agents, chemotherapeutic agents as part of the conditioning regimen that can have untoward side effects both in the transplant period, but over the next year following the transplant. And there can be the development of graft versus host disease where the graft attacks different organ systems in the recipient's body. Gene therapy, limitations of it's still under investigation. So there are no approved products for CGD gene therapy. These things are still being studied, and so there's going to be limitations just inherently associated with that. Earlier trials, and I mean much earlier, so a couple decades ago earlier, were associated with malignancy. And so there is always going to be that theoretical risk of when there is gene manipulation, are you going to cause a new problem? And so the FDA has a lot of regulations surrounding this to really prevent that from happening. But it did occur in the earlier trials, which were abandoned. Something that's not a limitation, but still it sort of is a limitation, but not, is you still do require chemotherapy for treatment with gene therapy, although be it significantly less than with allogeneic transplant. It's also sort of on the table whether gene therapy will cure inflammatory disease, and I'll show why in a minute. And then it's expensive. And so if we look at just the companies who are producing or developing these clinical trials and ones that have been successful in other for other indications, it really takes a lot of capital to develop these therapies. And then that comes at a cost once they are actually approved. Next slide. All right, so why gene therapy for CGD? So this is for those who are sort of science geeks, this is a nice little picture of hematopoietic stem cells. And what gene therapy does for CGD is it's offering a one-time definitive therapy for any genetic disease, but specifically for immune deficiency. And you can see down at the bottom in the green square, the myeloid lineage. So all granulocytes, which are the cells that are really affected primarily in CGD, start from a hematopoietic stem cell up in this like blue kind of trapezoid looking thing. So hematopoietic stem cells develop, and they develop into all of these different types of hematopoietic cells who then have long-lived production within the bone marrow. And so one could argue, and has been accomplished, that if you alter the genetic makeup of these hematopoietic stem cells, every cell that then is produced in progeny from those hematopoietic stem cells will have that correction in place, thereby sort of getting around the defect that's present in CGD. All right, next slide. Now, there's a lot of different ways one can administer gene therapy, and some of this is a little bit of vocabulary soup, but it's really important to think about it. So in the upper left, which is the blue square, is what I've just been talking about, allogeneic transplants. So a donor who is not yourself is identified, and bone marrow or peripheral blood stem cells are harvested from that patient and then infused into the recipient, which is this brown figure in the middle of the screen. When we think about gene therapy, there is ex vivo and in vivo approaches, and there are sort of gene addition therapies and gene editing therapies. So when we think about ex vivo, that's the Latin term for outside of the body. So the gene manipulation is happening outside of the person who is undergoing the gene therapy. So if you look at the upper right-hand corner, in red, in this case, they're using an example of beta and gamma globin, which is not relevant here, but it's really just trying to show the purpose of this. So a patient is going to have their cells removed from their body. Those cells are going to go to a lab, and the new gene of interest is going to be added to those cells, and then the patient's going to receive their own cells that have been genetically modified back into them. So they're receiving their own cells. They never receive anybody else's, and those cells have been manipulated in a lab away from their body. And that's really the concept of ex vivo gene therapy. Ex vivo gene editing is very similar in the sense that those cells are, again, removed from the recipient. They go to a lab and they undergo editing. But instead of the addition of a new gene, the gene of interest is edited, literally like editing a word in Microsoft Word. It's just changed. The alphabet is changed so that the gene is no longer pathogenic, but is actually functioning normally. And then those cells are, again, reinfused back into the patient. So the patient always receives their own cells back to them, and those cells have been modified in a lab. In the lower left-hand corner is in vivo gene therapy or gene editing. And what this is, so in vivo is the Latin term for inside the body, so inside the person. So unlike the last two examples that I gave you, the actual gene modification occurs inside of the person. So their cells are never removed from them. The vector that's going to provide the gene of interest is injected or infused directly into the person, and that gene acceptance, so I'm going to call it that, I'm totally making this up, but the gene kind of interest that's going to go into those hematopoietic cells occurs inside the recipient. So all of these technologies are being explored in CGD currently. All right, next slide. Click it again one more time, Colton. So just showing sort of a summary of earlier gene therapy trials. So this is the ones I was meaning were decades ago, where there was a lot of or associated malignancies. So these were two cases, one out of Frankfurt, one out of Zurich, where patients received gene addition therapy, and then developed myelodysplastic syndrome, and ultimately required bone marrow transplantation to cure their cancer. When this happened, the FDA, at least in the United States and the EMA in Europe, shut these trials down and said, go back to the drawing board. You guys need to figure out how to make this work without causing cancer. And that's really what people have been working on since. Next slide. The most recent sort of trial for gene therapy is lentiviral gene therapy for X-linked CGD. And there were several sites in the US and in Europe who utilized this study. And this is a lentiviral vector. A lentiviral vector really is a type of virus, and the virus is what introduces the gene of interest into those hematopoietic cells. And so this is just what this picture just is showing what the gene looks like. That's going to be introduced. Patients also received a targeted dose of busulfan, but a single agent chemotherapy, unlike the allogeneic transplant where they're receiving several agents. Next slide. So in this study, they treated around 9 patients. The results have since been published, but they did have a few bad outcomes. Click again one more time, Colton. So there were 2 deaths on the study. One in Europe and one at the NIH, related to one being hyperacute idiopathic pneumonitis, and the one down at the bottom was due to a cerebral hemorrhage that was related to a pre-existing fungal infection. And then the other patients did do well and survived the procedure. Next slide. This trial showed really, really good results. And you can see these are the patients 1 through 9, showing their neutrophil DHR. You know, many of you are familiar with DHR and how we diagnose CGD and measure superoxide capacity. And patients had therapeutic DHRs throughout the study. And then the bottom graph is really showing vector copy number being expressed within neutrophils, which was also decent amongst patients. However, if we looked at their clinical disease, 2 patients in this cohort had a recurrence of their colitis, thereby meaning their colitis got better, but then came back 3 to 5 years post-gene therapy. And in those 2 patients, they did have what we would consider a normal DHR for the purposes of a clinical trial or even for transplant outcomes of around 30%. And so this begs the question of whether at least this specific technology will lead to long-lasting cure of inflammatory disease specifically. Next slide. There also has been a new paper that's come out showing that inflammation within the bone marrow can compromise CGD gene therapy, thereby being what this really is showing is that chronic inflammation that coexists with the development of inflammatory disease or chronic infections can compromise how gene therapy sort of engrafts or takes or is successful. And how do we combat that, not just for gene therapy, but also transplant to prevent any failures? Next slide. All right, now I'm going to talk about 2 of the sort of ones that have been in the news lately, or you probably have heard about through various venues. And in both cases, the companies for these did provide some information for me, so I'm sharing their information that's been made public. So prime editing for P47 Fox CGD. So can you just click all the way through, Colton, so it's not animated? Perfect. I think there might be maybe one more. Oh, no, I told you wrong. I'm sorry.
No worries.
There you go. Okay, so prime editing. So this is Prime Medicine is gene editing technology that's really programmable for both search and replace. So this is that gene editing technology that I described and is specific for patients with P47 Fox CGD. So their technology allows for the localization or the searching specifically of the break in the P47 gene, which is NCF1, or the deletion, 2 base pair deletion in NCF1 that causes P47 CGD, and then edits in basically a replacement copy of that so that it's able to function normally, leading to gene correction. Next slide. They had to produce a ton of preclinical data to ever get this trial off the ground, showing, safety, showing on-target effects, that they're directly hitting NCF1 and not any other gene, and that they're not leading to other genetic abnormalities by infusing this technology into a person. Next slide. And so this is the data. 2 patients have been treated with their product, which is called PM359. Patient 1 in blue and patient 2 in pink. And in both of these cases, they had really good DHR positivity within 15 days of infusion, and tolerated conditioning just fine. They had rapid neutrophil and platelet engraftment, really conducive or in line with what we would see from allogeneic transplants. Rapid recovery of NADPH oxidase activity as shown by the DHR. And then patient number 2 has shown improvement in their inflammatory markers. This patient had colitis, and so some of the markers of that colitis have improved substantially post gene therapy. Next slide. But many of you have probably seen this New York Times article, which came out just this past week, showing and really highlighting patients who did receive PM359, who were cured, but now this product is not available and the clinical trial has closed. And so that's me. That's awesome. It's a great picture.
Welcome, Tracy. Thank you so much for joining us. We have a patient with us who has gone through this trial, so that's very exciting.
That's awesome. Maybe you can speak about your experience, Tracy, when I'm done, if you want to chime in. I only have a few more slides. But at any rate, in this article, they detail how really the burden to get these therapies, get these trials completed and get these therapies to market is extremely difficult. And like I said at the very beginning, very expensive. And so as a community, I think we can spend some time when I'm done talking and maybe when Tracy speaks to talk about how to advocate for this and how to get our regulators to pay more attention to rare disease and understand the nuances of these clinical trials. I can tell you that this company has not given up on this therapy. They are engaging with the FDA to try to figure out how to bring it to market the best that they can. So a lot of very nuanced conversations that have to happen. Next slide. All right, the last one is insomas. So insomas in vivo gene therapy for CGD. So this is for X-linked CGD, and their technology and their program is not at all the same as Prime. So look differently at these next few slides as well. Next slide. So insoma is potential in vivo gene therapy. So remember when I showed you the different types, this is in vivo. So this is a gene therapy that's not going to remove a patient's cells. Those cells are not going to go to an external lab. The gene therapy is happening inside the patient. So this is really a novel approach to gene therapy. And it's a process that includes enrichment of genetically modified cells. So what's going to happen is hematopoietic cells receive a viral-like particle that delivers genes of interest. And in this case, it's the CYBB gene, which is the cause of X-linked CGD. And then that leads to functioning neutrophils. Those cells that have received this viral-like particle will then home to the bone marrow and populate the bone marrow and produce NADPH producing neutrophils. NADPH oxidase positive producing neutrophils. Next slide. So this is just showing the schematic. This is the treatment journey. And just to remind everybody, this isn't a clinical trial. This is not available at all your institutions. This is, like I said, under clinical investigation. So a trial participants identified agrees to be part of this, and they will undergo a mobilization phase where that leads to an increased number of stem cells in the peripheral blood. And that is not in order to harvest those cells. It's so that there's a larger number of cells that have the opportunity to receive the viral-like particle once that's infused into the patient. So the patient will receive that. They will undergo this enrichment phase where they do receive some chemotherapeutic agents and some immune suppression to control how these cells sort of behave and don't kind of go crazy in the midst of this, and do sort of behave the way we want them to, go to the marrow, do what they're supposed to do, and then come out on the other end with a normal DHR. That is the goal. Next study, or I'm sorry, next slide. So this is just their clinical trial design. It's a phase 1/2 open label study that's lasting for a year. There will be a very long follow-up period, as is required by the FDA. The study population is initially going to be adults, but will quickly open up to pediatric patients. And they are currently open at the University of Minnesota, but soon to open at 8 additional sites, including my own. So we will be excited to be able to treat patients with this. Next slide. Okay, and then just to be balanced, there are other open trials for gene therapy. These are really available at the NIH. And I've listed just the clinical trials ID numbers. One includes technology called base editing, which I didn't touch on today, but is just another type of editing for gene therapy. And then another lentiviral vector study, or I'm sorry, 2 lentiviral vector studies that are available as well. Next slide. And then just I'll end with, again, kind of the regulatory hurdles. Click one more time, Colton. Are enormous. And so just to keep that in mind, why this is taking forever is because it really takes forever. So in order to ensure efficacy and safety and meet the regulatory requirements, it just is a monumental sort of Herculean effort. I also remind patients or potential patients that the FDA requires monitoring for these studies for 15 years. So when you enroll on one of these studies, you are really committing to it, to being a research participant for a long time, and to be followed for a long time following that therapy, which I agree with and should absolutely be done, but it's definitely a big commitment. And I think that might be it. Go to the next one. Yep, this just thanks my collaborators, especially the PIDTC and a lot of people I work with. And I'm happy to take any questions.
Thank you so much, Dr. Leiding. And we also wanted to thank Ensoma for sponsoring this talk. Thank you so much. And thank everyone for attending. And to your point, Dr. Leiding, the article in the New York Times, that was September 22. I hope everyone got a chance to read it. And we also have Mr. Attenbury here today, who was featured. He has his picture in the New York Times. This is really CGD's moment. We cannot believe it. Actually, you know, when we think back where we were back when my son was diagnosed, it was really not on the radar the way it is today. So it's really a testament to your work, a testament to our community, a testament to the CGDAA and everything that we're doing to put a spotlight on CGD. We also had a listening session recently with the FDA. We're doing what we can. But back to what you said, Dr. Leiding, which was really relevant, and what we can do. The article in the New York Times seems to ask, what is possible? What is the next step? And where can we as a community get involved? And so wanted to talk to you about that as the first question, then turn it over. What can we as a community do to help these treatments get to the finish line?
Yeah. I mean, I think there's a few avenues to pursue. I think the FDA is learning as these trials are being performed. It's, you know, it's a steep learning curve sort of on both sides of the fence. They've done a lot to treat their review process differently than for any, you know, standard drug therapy, like your blood pressure medication or your cholesterol lowering medication. But in some respects, they haven't. And they really have to think of gene therapy for genetic diseases as a completely different entity. And I think that really that separation and making sure that holds true is really important. I think they really have to think about rare disease differently. This sort of concept of we're going to enroll, you know, 50 patients is just not realistic. They need to understand that enrolling a few and showing excellent results in a community that otherwise doesn't have great options is okay. And perhaps the data that convinces them of a full approval happens in a phase 4 setting or out once it's sort of to market. And lastly is, and we did discuss this on the FDA call, and I think is really important, is making sure that the FDA understands the natural history of CGD and what a cure really looks like. So Is that going to be a resolution of an infection or the prevention of an infection? What does that look like for an individual patient? And it may not be the same for everybody. And we are unique in the sense that we have a way to test disease activity, and not many diseases have that. And we need to really drive that home with them so that they understand we can utilize this as a marker of disease. And it will help significantly with trial design and helping these entities, companies, academic institutions, whoever is pursuing these approvals to get that done.
Thank you. And the FDA has welcomed our input. We're so grateful that they did invite us to have a listening session. They heard us. We had a panel of patients with CGD who talked, caregivers as well. And after this call, if anyone would like to be involved in this advocacy effort, we're thinking about different ways to help continue to have our voices heard with the FDA, to continue to help ensure that these treatments get to the finish line, from letter writing campaigns to other advocacy efforts that we can make. I really exhort everyone who's on this call who'd like to be involved to email me personally, because we'll be working on that in the coming months and years, I'm sure. Okay, to turn it over to our guests. Is there anyone who has questions? I think that Colton is another one that you might know here. I'd like to introduce Colton Teicher, who is our communications consultant, former CGD patient, also went through curative therapy himself. So as you know, the CGDAA, we take pride in standing behind what we do. Is there a question, Colton, that you saw in the chat from our community, or would anyone like to raise their hand and ask a question, Dr. Leiding?
I don't know. Did Tracy want to say something first?
That would be great.
You're willing.
If he wants to, yeah.
There he is.
Hi.
Tracy, welcome.
Thank you. I don't know if I have anything to say. I can answer questions as to I'm at about day 120. Right now, I don't know if you I'm at the Sapphire Lodge right now, actually. This is the library room behind me. My DHR from this morning was 83.7. So, which is pretty good.
Great.
I'm pretty happy with that. The inflammatory bowel disease is improving, as Dr. Leiding mentioned. That I have charts and graphs, but that seems to be looking a lot better. Early days on that, like I'm at 120. That bears a lot of continued monitoring, but that seems to be improving as well. So far, so good. Maybe at my 6-month visit, we can remove some of the meds that you always have to take. We'll see. But so far, so good.
Well, that's so exciting. Thank you for joining us, and thank you for having your voice heard and being featured in the New York Times. I
hope that helps. I know it's difficult to get successful therapies to patients because of all the financial issues involved. So I hope that exposure helps a little bit in
it
nudging
absolutely helps.
somebody.
Only because of courageous voices like yours, Tracy, that we are getting our voices heard as well as a whole. And we're seeing more attention on CGD than ever. And that means that I hope there are going to be better, safer treatment options, which has always been our goal at the CGDAA, to offer these options to our patients and educate them. Well, we're not offering them, but educating our patients about what is out there for them to consider when they're making those decisions. Do we have any other questions for Dr. Leiding or for Tracy?
I think there is. I see one in the chat about prerequisites for gene therapy, a patient not having a previous transplant. And I have a couple of reasons for that. One is the confines of a clinical trial are not the same as the confines of using a therapy after approval. So any clinical trial is going to have very tight inclusion criteria and very tight patient population characteristics in order to be able to prove and to show to the regulators that something is effective and safe, and limiting variables that might influence that. So that's one reason. The second reason is that depending on the outcome of that transplant, there may be more than one immune system present, and maybe more than one genetic exposure or a preference for CGD present in the marrow. Meaning, if a patient is part donor and part recipient, then they're going to have some cells that have been replaced by a normal gene, depending on the type of CGD, and then some cells that are still expressing the mutated CGD. And at least with how gene therapy stands right now, it's very difficult to sort of predict which cell is receiving which. The third reason is chemotherapy exposure. So you have to remember you're also exposing again to more chemotherapy post-transplant, so that has to be taken into consideration. It's not certainly a deal ender, it's not too much chemo, but it does have to be thought about for any given patient. So I don't want to say that it means in the future when these approvals come to, or these products come to market, that it can't be considered in somebody who's had a transplant before, but these are the reasons why those confines are often put in place for a clinical trial.
But that doesn't mean, for example, once these treatments are approved, that patients who might have not been eligible would not be able to take them after they're approved by the FDA, or what does that mean?
It depends on how the approval comes about. So if the FDA excludes those patients with that approval, then yes, they would not be potentially eligible. But the FDA rarely does that because they want to leave that decision into the hands of the treating physician. And so they really only tend to exclude patient populations if there's a safety concern. So if they were to not exclude patients who had previously undergone transplant, then that would be a conversation with your physician of whether that's the right decision, given the circumstances of that previous transplant.
And so this is another opportunity where, Dr. Leiding, as you've said, there are these areas that are so new for patients and caregivers who have gone through transplant who might not be eligible. This is an opportunity to also have your voice heard after hopefully these treatments are approved, to be able to talk and make sure that if there is an opportunity, you might have the right to try it. So we just know that there are many caregivers and patients who have had this question. Perhaps they've had transplants that didn't work, and they have expressed a desire to be eligible. They know that they're not, but would like to think if it is ever approved, I might have that opportunity at some point in the future. And this is at the time when CGD is in the spotlight, and there's still an opportunity to work with the CGDAA and talk about these issues and make sure that you have a seat at the table. So people have been asking me for my email address. Colton, could you please put my email for everyone on this call? If anyone who would like to be involved and advocate and make sure that we're getting our voices heard with the FDA and with all the stakeholders, this is the time to do it when CGD is really in the spotlight and making big news.
So there's
Yes,
a couple
absolutely.
more. I can see the chat, Colton. There's one.
Okay.
Is there a reason why highly lionized female carriers are excluded from most gene therapy trials? Is there a timeline on when females will be considered included in gene therapy trials? So, it depends on the degree of lionization, but I think in most physicians who treat CGD's opinion, a female that is highly lionized to express the mutant gene is no different than a boy with X-linked CGD. They're really one and the same. They have the same kind of disease course and disease activity. And there have been successful bone marrow transplants for those girls or women who have had highly lionized disease with disease activity. And so I do think there's opportunity there for those women to potentially be candidates. The question is how much disease activity warrants something like gene therapy or transplant. Remember, I showed you data, at least from the transplant world, of 1 in 5 are dying from that procedure. So that's not to be taken lightly. That's not the same case with gene therapy. They've had excellent survival, but it's still a very big process. So it's sort of unclear at this point who and when for females, but I do think that those who are extremely lionized, it can be argued that they are really X-linked CGD from a genetic perspective. The second question I see here is if FDA approves gene therapy, will it be for both companies or just one, meaning in body or out of body? I think Insoma's trial is just beginning. They have not yet applied for approval. They've applied to perform the study, and they have approval to do that. But they have not applied for the approval of their product to then go to market. So they're, a ways away from that happening. They're going to need people to participate in the clinical trial. So I think that's where you can help them the most. For Prime, their program, what they've announced publicly is that they have closed that program and are not intending to treat any more patients on the clinical trial, but they are engaging with the FDA to see if there's a way to still make it available for patients in some capacity. So I don't know what that exactly means, but that's what they're working on. They're not abandoning the community or obviously the great results that they've seen thus far with their 2 patients that have been treated. So, but they are separate entities, totally separate processes, totally separate journeys for their path to approval.
Right, and for separate forms of CGD, Prime is for autosomal recessive P47, and Insoma, the clinical trial that just opened, is for X-linked CGD. And so, any other questions that we have? We have 8 minutes left.
I have a question.
Sure.
Are there side effects that may not be associated with an ex vivo gene therapy that may be associated with an in vivo gene therapy? And may we not even know about them at this point?
Yeah, I think the major side effect is not really related to the infusion of the products per se. It has to do with apheresing or collecting of cells. So in an in vivo approach, there is no collection. Nobody has to harvest marrow. Nobody has to do a peripheral blood stem cell collection. And so you avoid that entire procedure. And there can be complications associated with that procedure, although be it in CGD, it's not a common problem. It can be a common problem in other diseases, but not necessarily in CGD. And it also, the major benefit, I think, from an in vivo versus ex vivo approach is you get around the need for the cells to go to a lab and undergo sort of the genetic manipulation in that lab, which really is cumbersome and costly and has tons of regulation surrounding it. Whereas if you're administering the therapy directly to a patient, all of that is happening within the patient and can be monitored, but it sort of bypasses that whole other arm of the therapy. But from a medical perspective, as far as safety, that's not been shown. They're obviously under investigation, so not all of that is out, but there's not really enhanced theoretical risk.
Thank you, Dr. Leiding. And we have a lot of questions from our community about overcoming the hurdle of really understanding how a clinical trial works, where they can go, how they can find out more about it. Certainly on our email newsletter on our website, we educate our community. We talk about clinicaltrials.gov, but the site itself can be a little challenging to navigate. What is an easy way for our community to learn more about the clinical trials that are available? And also they have questions about where are they? Where are they taking place? How can they get to them? How can they travel to them? Those sort of basic logistical questions can be very overwhelming for our community. So if you could talk us through that in our last few minutes, that would be helpful as well.
Sure. I mean, there's multiple avenues, and I think you named a lot of them. One is contacting these companies directly. They're happy to direct you to where the trial is open. One is physician referral. Really, your physicians who are taking care of you should know this. And if they don't, they should figure it out and tell you. And I would really advocate that you push them to do so. Thirdly is obviously Felicia's group. She publishes these newsletters. She's going to let folks know when sites open. And then lastly is clinicaltrials.gov, which can be a little cumbersome to use, but it does list sites on clinicaltrials.gov to know where to go. And honestly, you can just Google these sites and sort of find them. But in general, it's really any avenue to find where to go. Now, what that means, you can obviously go see, I'll use myself as an example. If as we're opening the Insoma trial, when that happens, and I may receive referrals directly from patients, I may receive referrals from other physicians, I may receive referrals from my own patient population. And in all of those, what happens is a patient is evaluated. They're told about the trial. They're told about, you know, the different criteria of the trial, what that's going to look like for that patient. We review whether a patient is really eligible for the trial. If they are eligible for it, and that's very dictated by the protocol, the clinical trial protocol, and the FDA. If they are eligible, then ultimately it becomes their decision of whether they want to participate in it. Once that happens, then we activate that and really get the patient kind of moving towards receiving that therapy. So that's what would happen, you know, if a patient showed up for that. But the way patients come to me might be from different avenues, depending on how they hear about the trial.
Exactly. And so at the CGDAA.org, people can find out about the clinical trials and calling the companies, and also going on clinicaltrials.gov. And one of the slides in your presentation talked about different types of research that is going on. And we had a question about whether or not there is machine learning or AI that is being used in any of these clinical trials to help speed the process so that we can get these treatments to the finish line for CGD. Is that at all coming up now as
Not that I know.
Okay.
I'm also probably way too old for AI and these machine learning things, but no, there is, you know, we do use, and I will say clinical trials in general, infrastructure of clinical trials is starting to really use machine learning for data, because this is massive amounts of data and how to analyze that data. But for the recruitment of patients and the development of the trial, not that I'm aware of, has anybody used AI or machine learning for that creative piece of this. But I think for the data analysis, it certainly could be a useful tool.
Well, we definitely have seen, to springboard off this question, the FDA also released news last month that they are looking to make, to simplify the process for rare disease clinical trials. They're really working on this, but I do think they continue to need our help. That's why they accepted our proposal right away to speak to them. They had said that they get inundated by a lot of organizations that like to speak to them, but they were very eager to talk to the CGD community because they really do value our input. And so what we can do after this call is we've been thinking about different ways that we can reach out and different letters that we can write. We have different things that I've thought would be helpful to talk about how we really want to be heard and how important this is. We do want, as a CGD organization, to know that there are safer and there are more effective treatment options out there for people who do want to go the curative option route. It's up to everyone on our call today and throughout our community to decide. I always advocate for doing the decision that is best for you. We're here to provide the information and to educate, but ultimately the decision is yours. And so if you do decide to continue to think about this issue and want to advocate, we are planning some things that could be very exciting in the future. And in terms of other things we'd like to plan for the future, Colton put the fundraising tab in there. We will have fundraisers at the end of the year for Giving Tuesday, Christmas, Thanksgiving. We can't do this without your help. If anyone on this call could donate even, you know, $5, we would appreciate it. So the link is there just to show that your voices are heard with us. We do appreciate your vote of confidence in us and your support. So that brings us to 9 p.m. Thank you, everyone. I look forward to being in touch with everyone after this call for any questions. You know where to find us. We're happy to relay any other questions that you have to Dr. Leiding, and have a good night. Keep in touch, everyone. Bye for now.